Okay. So, okay.
Hello, everybody. A very good afternoon to all the participants. A very good afternoon to Dr. Parul Singh. She has recently joined us and she's trying to connect with the screen. I was just telling you that we had our first lecture on 3rd July. This is going to be our second lecture in the second international webinar series. The third lecture for this series will be going to be held
Okay.
on 10th of July.
Okay, fine. I'll start now. Okay.
The
Okay, fine. Bye.
speaker for today is Dr. Parul Singh, who is postdoctoral fellow in Aspili Lab, Collège de France, Paris. And the topic for today's webinar is Understanding Stress Response Adaptation in Adherent Invasive Escherichia coli. I'm Shomini Parashar, Assistant Professor in the Department of Biotechnology, IMS Engineering College, Ghaziabad. And on behalf of the entire department, I welcome Dr. Parul Singh and all the inquisitive participants. They generally make the event very successful. So a warm welcome from on behalf of the department to all of you. Dear all, I would like to tell you that the Department of Biotechnology at IMS Engineering College was established in the year 2002 with an undergraduate engineering program in biotechnology. In 2010, MTech was introduced to cater higher educational requirement of students. Since then, the department has been actively engaged in research activities in various areas of biotechnology and related fields, including research projects of UP Pollution Control Board and malaria vaccine development. Our department is NBA accredited and our college is grade A NAC accredited. Our department is also an authorized research center for PhD through Dr. APJ Abdul Kalam Technical University, Lucknow. We offer 92% placement to our BTech students in various biotechnology, biotech companies, as well as non-biotech companies. So this was a brief introduction of our department. Dear all, we are very thankful to Dr. Parul Singh that she has recently joined us and she has spared time for giving the presentation. There are a few instructions for you as in the beginning of the presentation I told you, but there were few people. Like during the presentation, please keep your audio and video off and do not present your screen. Another thing is, like, people were asking about the certificate. So I would like to tell that you'll get the certificate of participation in this webinar once you click on the feedback link, which will be provided at the end of the webinar. And Mr. Priyaranjan Kumar is going to share the link after the webinar is over. And another question, like if you have any questions during the webinar, you can write down in the chat window, and that all the questions will be taken at the end of the webinar. So I would like to introduce Dr. Parul to you all. She did her PhD in Dr. Ashwin Rai Narayan's laboratory, NCBS, Bangalore in the year 2018. And she has got several international research publications in various reputed journals. Presently, she is working as a postdoctoral fellow in Dr. Olivier Aspili Lab, Chromosome Dynamic Center for Interdisciplinary Research in Biology, Collège de France, Paris. Her project title is Understanding Molecular Mechanism Responsible for Stress Response Adaptation of AICE, that is Adherent Invasive Escherichia coli, LF82. We are very much thankful to Dr. Parul Singh for sparing her precious time for us. The topic for today's webinar is Understanding Stress Response Adaptation in Adherent Invasive Escherichia coli. Thank you very much, Dr. Parul, and a warm welcome to you in this webinar. Now over to Dr. Parul for her presentation.
Thank you very much, ma'am. Am I audible? Is it the voice is clear?
Yeah, the voice is clear.
Okay. So yeah. So thank you so much for giving such a warm introduction. And yes, I'm very happy to present the work which I have been doing here as a postdoc. Just to give a brief introduction, like the name Adherent Invasive Escherichia coli, it's a bacteria, and why we are interested in this is because it is one of the microorganisms found in the patients of Crohn's disease. So Crohn's disease is a inflammatory bowel disease. As you can see, it's a chronic disease. By far, we still don't have any cure for this disease, and its cause are multiple and complex. It's known that due to dietary changes in people, due to immune system dysfunction in the intestine, in the gut of human beings. And there is also a very important aspect is altered microbiome. So let me just give you a brief introduction of what a microbiome is. So it's a sum total of all microorganisms present in our gut, and there are a lot of them. Most of them are beneficial and or neutral. Some of them are pathogenic. So AIEC LF82, which from now on I'll call the organism, it has been seen that in Crohn's disease patients, in the biopsy of the tissues from the intestine, it was shown that it is prevalent very highly. So why and how it is doing so is the interest of the lab and of my project. So what does it do? So as I said, the name is Adherent Invasive Escherichia coli. So it's a bacteria, but what makes it interesting is that this is able to adhere. So here what I'm showing you is the intestinal epithelium cell of, and if you see the yellow one, this is the bacterial representation, it adheres to the intestine. It's not only adheres, it is able to invade the intestine. You can see here, for example. And the most important aspect of this bacteria is not just it invades, but it passes through and gets into the macrophages. So just to brush up that macrophages are the very important part of our innate immunity system of human beings. And they are supposed to be able to kill the pathogens entering in the bloodstream in different tissues. But this bacteria not just invades the macrophage, it is able to survive inside the macrophages. And that is what is the important question we are trying to understand, how this bacteria is able to do that. The machinery, the macrophages, which are supposed to kill, it is able to not just survive, but replicate. And this might give us some clues on how this bacteria plays a role in Crohn's disease and how we can sort of in the long term find solutions and cure for Crohn's disease. So before I go into the question more, I will tell you like how with microbiology, with bacteria, how can one study? So I think that a lot of us know that there are a lot of diseases caused by bacteria. There's Salmonella typhimurium causing typhoid, Mycobacterium tuberculosis. So how do we study? How do we find out how these bacteria affect us, make us sick? One good thing about studying with bacteria is that you can easily manipulate them. So you can color code them to, like, if I have to say in lay language. For example, here in Salmonella case, the researchers added a plasmid with the GFP. GFP stands for green fluorescent protein. So you put a GFP tag and another dye, and you make it a switch on, switch off. So if you give certain sugars to this bacteria, it will give you certain color. If you remove that sugar, it will give you a different color. So given this kind of assays which we can do, we can then use this bacteria to put it back in the cell culture or in vitro assays. And depending on we can separate them, that how many bacteria are replicating, how many bacteria are dead, how many bacteria have just stopped replicating and they are just dormant, they are not dead, but if you remove the stress, they will be again active. So this kind of studies and these kind of plasmids, reporter strains have given us an immense tool to understand how these pathogens affect us and what are the different ways they affect us. So this is what it means by phenotypic heterogeneity. When a bacteria, a pathogen enters a host, it does not always has the same fate. It can die, it can be killed by the immune system, it can actively replicate, it can subvert the immune system. So these kind of studies we can do well by manipulating the bacteria in the lab and looking at how it does so. So here I show you another example of this study, that if a bacteria is not dividing inside an immune system, you see a different color, for example here, or else if it is dividing, it's multiplying. If you know, bacteria divides by fission, and so you know each parent cell divides into two daughter cells and so on and so you'll have a different color. And we have been able to understand a lot of these things in bacteria, for example, pathogens like Salmonella and Mycobacterium tuberculosis. But they present a small picture. There are many more pathogens, and we are interested in one such, which is AIEC LFET2. So here in the lab previously, it has been shown that, for example, if a bacteria is being phagocytosed is, let's say, eaten up by a macrophage. So you should remember that a macrophage, the job of macrophage is to kill the bacteria. But what happens that these bacteria either die or they activate their stress pathways inside them to become a non-grower. They stop, become dormant, and wait for that bad situation or bad environment around them to get away and then grow back. So this we realized in the lab previously, that bacteria is able to do this by sensing that there is less nutrients in the macrophage, and it is also getting a lot of DNA damage. So a quick word for bacteria, like in general, they are very adaptable organisms. They can sense presence absence of carbon sources and they immediately adapt, and that's what makes them successful. And that's what they are doing again here inside an immune system where they're supposed to be dead, but they are not. They are able to sense what is going to go wrong in their environment, and they change their genes, their transcription, their DNA, RNA, and protein in order to survive. And given this was the objective. So, okay, we understand that once bacteria enters macrophages in the immune system of host, it either dies, it can become dormant, doesn't grow, or it can actually behave as if nothing wrong is going on. It's another host, and there's nothing, no stress for it, or so it's actively replicating. So what my project deals with is to understand what makes it successful inside a macrophage or inside a stressful condition, because that will give us a clue of how bacteria are able to survive in stressful conditions in immune system. And also in long range, it's very important because nowadays we get to hear that there are multi-drug resistant bacteria. There are bacteria which are, which you get a relapse of infection when you think you're already all right. So all these things show that bacteria, when we think are dead or cleared by antibiotics, by the immune system, they are not. They are still surviving. They are still keeping it quiet. And when the situation becomes better for them, when you stop taking an antibiotic, for example, they come back. So that this in long range is what we want to understand. So how do we do this? As I showed you, like we can, using a bacteria, it's easy to manipulate. So what I did was, knowing that macrophages have lysosomes, which are very acidic compartments. So we thought that if we use an acid stress response gene from bacteria, use its promoter, tag it with green fluorescent protein, and then use it to track the fate of this bacteria inside, it will be an interesting observation. So I took certain different pathways, genes from bacteria. I use the same process. And I will not going to explain all of them, because for today's talk, I will talk about acid stress response, because we have some nice data to show like how it is changing and adapting in the stressful conditions. So what was the whole methodology, what we want to follow is like, for example, here we grow bacteria as a control without the GFP promoter tagged, and we add the green. For example, let's say this is what our constructs are. This is the LF82 with the green fluorescent protein, and we have a constitutive M-cherry fluorescent. So now here, to be clear, if there is acidic environment, you will have a green fluorescence. If you don't have an acidic environment, you will have red fluorescence. So red is constitutive, always present. Green is present only when there is acidic environment. When the bacteria senses, then it will produce that. So this is the in vitro studies. I'm going to show you some of the results, how we do it in controlled conditions in the lab. The other aspect of this, as I said, that we are interested in how it survives inside the macrophages. So for that, we are using monocyte THP1. It's a human monocyte. We do cell culture. We differentiate these monocytes into macrophages, and then we use the same bacteria which we have constructed with this green fluorescent plasmid. We infect the macrophages and track them to understand how the bacteria adapts or how it survives. So these two are very different strategies, and we want to use it in parallel to understand the molecular mechanisms of how this bacteria is successful in doing what it does. And once we have these two things, since I told you, like in a population, there is heterogeneity. Not all bacteria survive. Some of the bacteria are able to be dormant, stay dormant, not grow. Some of them grow, and some of them are dying. So we don't want to work with population level data. What we want to do is, for example, here, let's say a macrophage infected with bacteria, and you might have some bacteria which are producing GFP and some which are not. So we will sort them using FACS. FACS stands for fluorescent activated cell sorter, in which a laser, which you choose for your based on what fluorescent marker you're using, detects the cells, it divides them. So here, for example, I say I want high GFP population at one side and low GFP population at the other side. So what it means is that cells which are responding to the acidic environment, I group them together, that how they are different from cells which are not able to respond to the acidic environment. So with this tool, we are playing around, and we have a lot of different mutants which we are trying to work. But for today, we will just focus on this, if the bacteria is able to sense or not. And this whole pipeline we are setting, and then in the end, we want to do gene expression analysis. So what is gene expression? So we know central dogma, DNA to RNA to protein. There are certain exceptions, of course, but bacteria is very, very focused on producing the protein which it requires in that environment. So when we look at the RNA of that protein, of the bacteria, we will get to know which all RNA it thinks is important for that condition. And that will give us a clue and an evidence of how and what genes and pathways are important for bacteria to survive in the acidic environment, in macrophage, in stressful conditions. So this is the workflow which we are following. Now, as I said, RNA, once you get RNA, what kind of information you can actually get from bacteria? So I will show you again Salmonella. It's one of the most worked, researched pathogen. And again, here I show you some results of when you do RNA extraction and sequencing, the next generation sequencing, what you get. So just to give you a brief pathogenic history of Salmonella. Salmonella is a gut colonizing microorganism. It has Salmonella pathogenicity islands, SPI2. So these islands, initially it was not known very clearly how Salmonella is able to be successful. It's able to cause diseases. And they found these genomic islands. So for example, here you see like it's the genome, and these are the islands, genes, which were able to give virulence factors, the survival factors inside human host for the Salmonella to make it successful adaptation and create virulence in the pathogen. So this group showed that with RNA sequencing that whenever the macrophages engulfed Salmonella, and these Salmonella activated. So on the x-axis is the same location of this Salmonella pathogenicity island. I'll ask you to focus on this red and dull red line. So you see the genes are expressed highly. And what it showed that actually these genes are responsible for Salmonella survival inside the host. So similar kind of thing was done very recently for Salmonella, a different story. Here, they showed that the macrophages themselves are not very similar. So the Salmonella sometimes changes the macrophage environment. So for example, here, they took macrophages without bacteria, which has no bacteria, which is called naive. Bystander, the macrophages with bacteria, but there were no, like very few. Host killed the macrophages in which bacteria had all been killed by the macrophage. Non-grower, where macrophages had bacteria, but they were not growing at all. And the bacteria, macrophages where the bacteria were actively replicating. So these different classification, there are different kind of macrophages. Even in the same experiment, you get so much of heterogeneity. And what the authors showed that some of the macrophages which were able to kill bacteria had the classic inflammation signaling pathway activated. Whereas the bacteria where the macrophages where bacteria were able to grow well, they had somehow not activated their inflammatory pathways. The inflammation pathways are the ones which actually allow nucleases, caspases to come and kill the bacteria. So it seemed that there is some crosstalk going on between the pathogen and the host, and it's not a simple picture, it's a complex picture. So given this, let's come back. We know macrophages give a very hostile environment for the bacteria. There is less nutrient, it's an acidic environment when it fuses to lysosome, and there are DNA, RNA, genotoxic stresses. So how does the bacteria survive? What I did first in the lab was to first take some of these conditions and do an in vitro study. So for example, for acidic environment, I took the medium which we use to grow bacteria, a very rich medium, which is lysogeny broth. And I added concentrated HCl to a condition that the pH becomes acidic. Okay, so it's simple to do. And similarly, to get nutrient deprivation, I use a chemical called SHX, serine hydroxamate. What it does is it induces the bacteria to think that there is less nutrients, and then the bacteria goes into this mechanism which it has evolved called stringent response. If I may say, it's like fasting. So if bacteria knows that, okay, I don't have much food around, what I do, I don't waste my energy, my carbon and nitrogen on things which I would normally do. Now I will hold on to my energy sources. So that is called astringent response. So I have these kind of chemicals, I use them. How do I do it in vitro without macrophages in the lab? I grow the bacteria. I use a 96 well plate and put the bacteria with the growth medium. I can play around with the stresses which I want to. I was talking in the previous slide. I can add acidic, I can add stringent response activated cells, and then I use a plate reader to measure the growth of the bacteria to see how the growth is affected and how, since we are using a fluorescent marker with GFP, whether there is a change in the fluorescence in presence of stress or not. So this kind of simple setup will give us some very basic and crucial information. So now I will show you some of the results of these kind of stress induction experiments. So again, remind you, here we are looking for growth of bacteria, whether the growth of bacteria changes with addition of stress, and the fluorescence in terms of GFP, green fluorescent protein. So here again, the x-axis is the time taken for bacteria to grow. y-axis is the optical density. So this is the measure of the bacterial growth on the y-axis. And these are my different strains, the control. I think it's very important to lay stress on this thing that you, when doing such experiments, you always need controls. You need to show that your control is not changing much across, and that's what we see. So when I have an LF82 strain without the green fluorescent protein, the blue, it's growing well. And when I have PASR or promoter with FIS or promoter of Arakin, you see from this here, the growth is more or less good. So good, we are not perturbing the system a lot. And for GFP in the same conditions, so again, the axis is same, except for this y-axis now is the fluorescence measurement. So again, we see very minimal. And this gray is LF82 with promoter FIS, which I have not talked to you about, but this gene is known to be very high when there is nutrient upshift or there's a lot of enrichment of nutrients in the system. And since we are using a rich medium, this sort of proves that our promoter constructs are working well. But coming back to ASR and our stress conditions. So here on the x-axis is time again. Y-axis is what I have done now is I have taken the fluorescence and I have normalized it with the growth. So now what we are seeing here is the result of when I add acid, I put the cells in the plate, I let them grow for some time, I then add acid, the first stress at this time point. And after some time, I add mitomycin C. Mitomycin C is a DNA breaking agent. It's used in, it's very lethal. It's used in chemotherapies and it induces DNA breaks. So it's a high level of genotoxic stress. So when I do this, I now measure PASR. What happens to PASR GFP LF82? And you see the one in yellow. The yellow is LF82 PASR in acidic conditions. So you see induction, whereas all other strains are at very basal level. So it's good. In acidic conditions, we see induction of our construct. It's a proof of principle that our construct is working well. And very interestingly, even if we add a DNA breaking, a very high amount of stress, such as mitomycin C, we still see the induction if we have already, the environment is acidic. So just one more time, I'll repeat that PASR GFP or this gene, the acid stress response gene is active if the environment is acidic, even if there are other stresses present. And this is important for me because when I'm going to use this construct for macrophages, there multiple stresses will be present and I want my construct to work despite multiple stresses. So now we want to look in the microscope, how does these bacteria look? So as I said, the control is M. cherry, which is red dye, and it is always present. So if there is no stress in the system, you will always see M. cherry. So here it's a microscopic image of the control strain LF82 without the GFP in LB. And here, as you can see, they are, I think, doing very well for themselves. So here I have done the measurement. So for example, it's seeing, but for statistics, for seeing that what results we are getting are significant. To say something, it's very important to do some statistics. So how we do that? Here, each cell size is on the x-axis, and on y-axis, I measure the fluorescence of GFP versus M. cherry. You can, we use ImageJ and Fiji software tools to quantify microscopic images and do analysis. And there, for example, here, so if there is high GFP, you will see the scatter plot, the points shifting somewhere over here. We don't see that because there is no GFP in the system. It's a negative control. We see very low GFP or negligible GFP, good, because this sets the condition for when we do the experiment with our promoter construct of interest. So now I show you the same result for LF82 HU M. cherry. Now here, When in LB, you see PSR is in negative. It means there is no GFP, even though it's not being induced. The moment I put acid in the system, you see the fluorescence shifts to high GFP. It's not clean. It means that not all bacteria are responding to the acid, but majority of them are concentrated here. So just to give you a brief explanation of these plots, these are scatter plots which are density based. So the darker the color means more number of points or more number of bacteria are present there. So if you can see here, you see a lot of bacteria are now induced GFP, and yet there is a distribution. And here you can see clearly in the microscopic image of when I add acid to the system, a lot of bacteria which were previously you saw all red with M. cherry, they turn into green. They start expressing the GFP, which is good. Also, there are some red bacteria if you see. So it shows that there is heterogeneity in the response. It's not all or none. It's somewhere in between. Also, if I add, again, as I showed previously, I added genotoxic stress like mitomycin C, which breaks the DNA of bacteria, you still see the presence of GFP, albeit not as clean as the plain acid. So this gives us a very, very important information that bacteria LF82, for example, already is able to be present in acidic environment. I mean, and even in DNA damaging conditions, it can survive. So knowing this, we thought of playing around because these are things which we can do easily manipulate in the lab and understand and see. So I started manipulating the system more to see if I deprive them of the nutrients and give an acidic environment, how do these bacteria behave? Here, I again do the similar experiment as I showed you previously, time on x-axis, optical density on y-axis. I put acid and stringent response activating SHX. And you see, so for example, the blue curve here is LF82 in plain LB medium. It's growing happily, good. The moment I add, acid on this time point, the orange line. So the orange line, if you follow, is the one with only acid. The growth becomes quite slow. So what it shows that it takes this long for bacteria to adapt to the changes in the environment. It sort of stops growing and the environment around it is not hospitable enough, and it takes time to adapt to the acidic environment and then restart growth. When I add a nutrient deprivation agent, which is in gray, so here the environment is already acidic, and now I add a nutrient deprivation agent, and you see it actually doesn't make a big difference. The bacteria is already adapting, it has stopped growth, and hence it is able to show very similar effect to what we observe only in acidic condition. And similarly, we swap it, we see what happens. So we see that when we add SHX first, again, I will ask you to follow the orange line. The blue line is the control, it's behaving well. The orange line is with the SHX. So when I add a nutrient deprivation agent first, it's not very different. So blue and orange has slight change, but not a significant difference, okay? So bacteria is able to cope well with nutrient deprivation, whereas when we add the acid, which is the gray line, then you see a drop in growth. Then you see a difference. But the difference in growth is not as huge as over here, for example. So what it means is that it is also important that when the stress is being sensed by the bacteria, initially when it is just coming out of the lag phase, it is difficult for it to immediately recover from the stress. But when I add later, the timing of the stress is important because then it takes lesser time to recover. The recovery is not complete, but still it is able to recover. So it makes very, very important points for us to consider is that it's just not that you can throw the stress whenever you feel like. It's very important if we want to understand and study these bacteria, how they survive, the timing of the stresses if we are doing an in vitro experiment. So here I am showing you the fluorescence, what we see. And again, as I showed you, I show you here GFP by OD. So GFP is good, but it has to be normalized by OD because each bacteria presents plasmids with the GFP. So if there are less bacteria, there'll be less GFP. So you can see when we look at the fluorescence, this is the time when we add acid and immediate induction of the promoter. So it's a good news for us because for our long-term experiments, this shows convincingly that no matter what kind of stress combination we are using, this promoter construct is robust. It can be trusted for our when we use it in macrophages for its reproducibility and efficacy. So this is something which is very, very important for us. It's a simple point, but again, very important that bacteria is able to successfully survive DNA damage, nutrient starvation, as well as acidic conditions. And this gives us a clue of how it would be able to successfully invade the epithelium and be a part of the whole dysbiosis of Crohn's disease. So we have done this. So if you remember my methodology, the aim is to go further to see if we can sort these subpopulations. We don't want to look at a population level. We want to see the population which is actively replicating versus the population of bacteria which is not actively replicating in the presence of stress. And that's why we are using a GFP and cherry and so on. So now once we are set with our constructs, we start with sorting of these bacteria based on fluorescence. So as I showed you previously, so now we have these bacteria in macrophages as well as from our in vitro studies, and now we sort them based on GFP high or GFP low. So I show you some results of GFP, as I think my audience is students, so I try to put a lot of stress on the controls because this is something which is very important to remember, that whenever you are doing an experiment, I think, in my opinion, it's very, very, very important to have more controls initially when you're setting up a project, when you're setting up a research question, than your organism or than your interest test, because the controls need to be, this sort of solves a lot of reproducibility issues in general, which we find in our experiments. So a negative control means there is no fluorescence, okay, and NLB. So the result here, I'm showing you facts, is like this. So on the x-axis is green fluorescent protein, on the y-axis is the DAPI. So what is this? So when you do FACS, you have multiple window channels. You can actually sort cells based on size, density, granularity, fluorescence, etc., etc. We chose green fluorescence and DAPI. DAPI is a nuclear stain. This stain binds to the minor groove in DNA, and whenever it binds to DNA, you will see the blue color channel of this. So this FACS is very sensitive instrument. You can very quickly sort out large amount of populations with this and get results very quickly. But again, it's very sensitive, so you need to be very careful. So here we take bacteria, which is negative control, and we sort of put our measurements in a way that if you look at this quadrant means it's high in GFP and high in DAPI. There are no good. This quadrant means they are low in GFP and low in DAPI, and it is true because these points which you are seeing means that these are basal level noise from the cell. It is a very sensitive machine, so you will definitely see some points, which is okay, as long as it is not in your high fluorescence region. We then use a positive control, which is highly fluorescent, okay? So you see now, in this case, this positive control is high in GFP and high in DAPI. Good. So our gating, then we can decide which gates to put on these to sort out our population. So now we go to our test. We take our LF82 with PSR GFP construct and just in the medium, plain medium. And you see that they're high in DAPI because they have DNA, but they are low in GFP. They have not been induced. There is no acidic environment, and hence there is very basal level of GFP present. And now we use our construct of interest, which is PSR GFP in acid. And now you see the high GFP population emerges. This is what we are interested in working with, how this population is different from this population. These are coming from the same culture of bacteria in the same environment, how this heterogeneity is evolving. So this is what we are doing right now. And we have multiple different combinations and permutations, but I think this is important. So, okay, enough story about in vitro, but this is what parallelly also we are doing is to take macrophages and use the same construct over there, infect the macrophages. So what I'm showing you here is the result of a macrophage infected with the PSR LF82 for 18 hours. So then I extract these macrophages, I fix these cells, and then I sort them using the same mechanism, principles of FACS. And here you are seeing the result of these. Each point represents a macrophage. And each macrophage may or may not have bacteria. We don't know. And that's what we want to do with FACS. So here on the x-axis is high GFP going this way. Y-axis is M-cherry going this way. So you can see it's a spectrum. You don't have high GFP population. You don't have low GFP population. It's a continuous spectrum. Like macrophages are there with no bacteria. For example, this population, macrophages with less bacteria here, and macrophages with high number of GFP producing bacteria over here. So we are interested in all these different macrophages. What is happening? What decides whether bacteria survives or not survives? Okay. So just to make sure that, okay, we are doing FACS, but what we are getting out of this is actually what we expect it to be, we do microscopy, confocal microscopy. So here you see the results. So this, I hope it is clear. This blue stain, which you are seeing, is DAPI again, and this is the macrophage nucleus. And then you can see the green bacteria, the GFP high, and the red bacteria. So some of them are expressing GFP or not, but they are present almost in the numbers of 30 to 40 each macrophage. And you can imagine that, like in terms of the disease, if you think about that, these many bacteria are actively present inside the macrophages, and this sort of gives us a clue of how this Crohn's disease is a chronic disease. Like it is a long-term disease because these bacteria are able to survive and evade antibiotics and immune response by just sitting inside the macrophages. So also you have bacteria with macrophages with fewer bacteria. And for example, this macrophage, you see there's 1 or 2 bacteria, and maybe some of them could be dead even. So this is what we are doing, and we have been able to successfully sort these cells, extract RNA, and we are in the process of doing the transcriptome. But this is something very exciting for us to see. So this is again another statistical analysis of the microscopy data from macrophages. And I will just give you a brief, this is the quantification that how much of GFP and M-cherry we get in macrophages with less bacteria versus how much of GFP and M-cherry intensity we get in higher number of bacteria macrophages. And we find that the number of bacteria is very different. It is very, very similar. So what it means is that macrophages with higher number of bacteria, for example, here you see the green line. In this graph, this axis shows that higher fluorescence intensity. So you see a lot of variation, a lot of heterogeneity in the number of bacteria one macrophage can have, the number of heterogeneity a host immune system can have in terms of a pathogenic bacteria like AIEC. And this is why we do FACS. This is what, so that we can characterize each population separately to understand the mechanism. And that's what our aim is. So this is for today. And just to briefly recap what I have said, that we have made several promoter constructs to understand that how this bacteria sense, adapt, and survive inside hostile conditions. These bacteria are able to do that, what we believe, with phenotypic heterogeneity. We observe that heterogeneity in each and every experiment which we do, that it's not the whole population survives, it's some of the bacteria are able to survive, and that's what we are trying to pinpoint on how we do that. And now we are undergoing the RNA sequencing, so we will have hopefully some candidates to work with. And just before stopping, it's very important to remember that with the incoming antibiotic multi-drug resistant strains, which we often hear, and this kind of studies are very important. And I think a lot of, not just us, but a lot of research groups are now paying attention to understand how these bacteria can be quite non-grower or survive inside very hostile conditions to be successful. And these kind of studies will give us a gateway to understand and maybe hopefully make better therapeutics and better antibiotics in future to have less less effect on the patients and less dosage of these kind of harmful antibiotics. So with this, I will end, and I would like to thank, this is the picture of our courtyard of Collège de France here. It's a very beautiful place. If you ever get a chance, you should come. I'm very thankful to Espeli team, where they're very helpful for their feedback, their comments, and their help with different strategies, because I come from a very different background, so it was easy to set me up here. Marie Agnes and Ivan Matik, they are the PIs in different institute, and they have been very helpful in their feedback and comments on the project pipeline. I would like to thank the Collège de France fax and imaging facility where I do these experiments, and CIRB and ANR for my funding. And I'm thankful to Department of Biotechnology, the faculty members of IMS Engineering College, and the students. Thank you for your patience and your time. And I am done. Thank you very much.
Thank you, Dr. Parul, for the wonderful and for sharing the research presentation with us all. Thank you very much for that. And now it's time to see the questions. Let us see what are the questions in the chat window. If you have any queries, dear participants, you are free to write down your queries in the chat window. There are many questions regarding the feedback. Is there any question? I'm not able to see any questions. So everybody is saying thank you, ma'am. Thank you for the nice presentation. See, I'm not able to see any questions. If you again, if you have any questions, please see to it that you write the question in the chat window. Okay. Okay, there is a question. Sneha Singh has asked a question. Could you please throw some light on efflux? Oh.
Hello.
Yeah. Hello.
Yeah, the question is, could you throw some light on the efflux? I couldn't hear the last part. Efflux pumps, is it?
Sneha, can you please write the question again?
Okay.
Yeah, pump.
Yes, so efflux pumps. So, with the acidic environment, I think it's a nice question, because with acidic environment, like usually bacteria are like the Escherichia coli are neutrophiles, so they shouldn't be able to grow with, but the gut bacteria, the microbiome, which I said that this is already present in the microbiome in the gut of human host. So these bacteria have adapted to the acidic environment, because if you remember that gut lining is acidic. So it's the efflux pumps in general, specifically, they are not, but I can say that, for example, if you have heard of EVG, AS, and these are the gene two component efflux pump systems which are very active. And if you delete, knock out these efflux pumps genes, the bacteria dies. The bacteria cannot survive in the acidic environment. So they are very, very important part of its survival in acidic environment.
Yeah, another question, ma'am, is from Kinjal. She wants to know that how common are E. coli outbreaks?
So E. coli is it's a very huge phylogenetic supergroup. So the E. coli which we normally see is the commensal, but there are different phylogenetic groups which are very, very common. For example, in India, you have EPEX, enteropathogenic E. coli, you have enterohemorrhagic E. coli. There are very common diarrhea and dysentery, which is caused by them. And for example, a lot of uh, infant and child usually contract these kind of E. coli, pathogenic E. coli infections, and are affected by them. And India is actually, it's one of the highest causes of dysentery and at times mortality in younger infants from the E. coli, pathogenic E. coli strain infections. So, and a lot of them is through intestinal epithelium infection. So that's why it's quite important to understand this. So it's very common in tropical countries. In India, it's quite common. And there are diverse strains of pathogenic E. coli which are present. Shigella, Salmonella, they all cause dysentery and diarrhea, and it can lead to very critical condition also.
Yeah, thank you, ma'am. Another question from Gulnaz is that which method is easy, in vivo or in vitro method?
So yeah, I mean, like, I think easy is it's subjective, because it depends on what kind of information you want to get. So let's say if I want to look at combination of stress, it's easy for me to do a macrophage experiment. You have to learn, so you'll have to invest some time in learning the techniques. But if you have time, I think in vivo experiments give you much more information in terms of actual host environment. But if you want to sort of focus on effect of individual stress agents, if you want to pinpoint what leads to affect growth of let's say acidic environment, we can do in vitro experiments, because here you can do multiple combinations in a very quick time. So I think it's about timing. In vivo experiments take longer compared to in vitro, but I think easy is, yeah.
Thank you, ma'am. Another question is from Sneha again. Can E. coli survive in deleted MFS efflux pump?
E. coli can survive without the efflux pump. Is that what? Uh,
Deleted, I think discarded efflux pumps she's talking about.
so discarded, like for example, in the strain which with I am working here, it is the acidic two component system is very important for it to survive. And if it cannot survive in acidic condition, if we knock out or delete the genes which transcribe for these efflux pumps, it cannot grow, it cannot survive in acidic conditions. In normal conditions, it can, but if the moment I add acid, they will all die.
Okay, we have another question from Hussein Jafri. How common E. coli characterization?
How common are E. coli characterization?
Yeah, maybe.
Okay, so maybe I think I didn't understand. I mean, like, is it to ask, can we, like, characterization
Maybe
for
how
E.
often do we characterize E. coli? How often do we characterize E. coli?
So characterize, yeah, like I think I'm still like maybe I'll just give a generic response if it I hope it is it's what he's asking he or she. So E. coli characterizations, I I would say that, I mean, like, for example, E. coli is very diverse. Like, E. coli generally does not cause a disease, like, like, but there are different phylogroups. It has evolved, it has grown very closely with the human host, that it has evolved in multiple roles. And at times when the host immunity is not very strong, if host is immunocompromised, some of these E. coli become pathogenic. So, I don't know if this answers this question, but yeah.
Yeah, I think all the questions are, there is one more question from Kinjal, I suppose. Ma'am, why are some E. coli infections resistant to antibiotics?
Okay. So that's what is like is becoming a leading cause of worry for medical professionals and researchers. There are multiple reasons why antibiotic resistance occurs, and I mean, like, there I can list a few. So, for example, E. coli, as I said, is extremely E. coli or bacteria in general are extremely adaptable organisms. You throw them in a new condition, they will adapt. As you saw, like if I'm putting them in stress condition, also they adapt very quickly. They take their time and they will adapt. So, what happens if in terms of antibiotic usage, a lot of places either people don't complete the dose, for example, in some cases, like, this is not my expertise, but this is what we understand, that, for example, if a doctor has given you 7 days dosage, but you take 5 days dosage and then you feel better and then you stop it, and that doesn't mean that if your symptoms of disease are over, it actually means that the bacteria are becoming less, they have not disappeared, and there are few of them still remaining, and you stop it. And what happens is that these bacteria have seen the antibiotic environment. They use drug efflux pumps, they use horizontal gene transfer to acquire the resistance to those antibiotics. And in my case, for example, we are also showing that it's not even requiring an efflux pump, or it is not even requiring mutation. It is able to do it, but just being different from the rest of the bacteria. So there are multitudes of reasons why antibiotic resistance happens, but one of the very important is that horizontal gene transfer. Bacteria realizes that I don't have resistance to this specific drug, but somewhere nearby there's another bacteria which is surviving, so I will acquire. All other ways is by using efflux pumps, by removing the bacteria from themselves. The others are like being collective, living. Like, for example, some bacteria form biofilms, and in the biofilms, it's very difficult to penetrate the drug or antibiotic, and hence uh, they can survive drugs and all. So it's very important to follow the course of the drug, whatever is being said, and uh, like, yeah, not taking very high dosage of drugs. So there are, I think it's another seminar for how antibiotic drug resistance happens to bacteria, but it's very, very important to keep control the dosage and the period of your antibiotics.
Yeah, ma'am. Thank you. Another question from Sati Mittal. She asked that what is the best treatment for E. coli?
So again, like, let's not use E. coli, because E. coli is, I think, beneficial and not harmful. But these invasive or pathogenic strains like exec, EPEC, EHEC, AIEC, here I think it's very important, if you have these infections, to go early to the clinicians, to your medical practitioner, and use the drugs, because Crohn's disease is right now uncurable. You can reduce the symptoms of Crohn's disease, but you cannot cure it. It's a chronic disease. But a lot of patients, by taking good dietary choices, probiotics, and being with the following the drug regime which their medical practitioner is giving, can live along with the Crohn's disease lifelong, and it doesn't affect their life very much. But other diseases like diarrhea, dysentery, and other gastroenteritis, for them, I believe that there are already efficient drugs available, which, yeah.
Yeah, Mr. Mohanty has asked a question like, are all varieties of E. coli become resistant to antibiotics?
No, that's what. Like I have been like the common human commensal E. coli is not pathogenic. It's not drug resistant. There are other strains of E. coli which can be pathogenic and drug resistant. And as you saw in my own presentation, that it's not that all the bacteria are able to survive. Some of them are able to survive. And that's what we are trying to understand, because this is one of the pathobiont present in Crohn's disease, which is known, like which is etiology and mechanisms are not yet known. So that's what we are trying to understand. But bacterial diseases, for example, for Mycobacterium or Salmonella, it's very, very clear that, like some of them are drug resistant. There is emergence of drug resistant bacteria. It's not true to say that all bacteria are drug resistant, but there is in recent years emergence of drug resistance bacteria in general.
Is there any protein essential for bacteria survival? It's a question by Swarada.
Is there any
Protein essential for bacteria survival?
Okay, so bacteria, I think different studies have shown that there are some genes and some master regulators and transcription regulators important for bacterial survival. So, for example, there is, as I was mentioning, stringent response. So you have this gene called DKSA, which tells bacteria that, okay, right now there is less nutrients, right now there are more nutrients. So it's a very important protein. It's a very important gene for bacteria. They are acidic, like as I said, the efflux pumps are very, very important. There are genes which help bacteria repair their DNA and DNA, which are very important again. So it's not one gene I can say, but there are a lot of processes like information processing pathways of bacteria, the metabolism pathways, and the acidic efflux pumps pathways, which are very crucial for bacterial survival.
Thank you very much, Dr. Parul, for patiently answering all the questions of the participants. Thank you once again. And now I would like to invite our HOD, Professor Rashmi Chandra, for vote of thanks.
Thank you, Shomini. Good afternoon, all of you. I, Rashmi Chandra, on behalf of Biotech Department of IMS Engineering College, Ghaziabad, extend a very hearty vote of thanks to Dr. Parul Singh to delivering a wonderful talk on understanding stress response adherent invasive E. coli. I am very much sure everyone must have gained knowledge on the topic. Thank you, Dr. Parul, for gracing us with your presence and taking out your valuable time. I appreciate the delegates from different countries, students, professors, and faculty for their participation. Also, I would like to extend my special thanks to Mr. Priranjan Kumar, Ms. Neha Singh, Ms. Romni Parasar for arranging such a informative lecture. Last but not least, I extend my big thanks to our whole department, including our bright students and each and every faculty member for their support. Thanks for making this talk a successful event. Thank you. Thank you very much. Take care. Have a good day. Thank you, everyone.